Systemic mastocytosis is an uncommon disease, but patients really have a very difficult time, specifically in the aggressive phenotype. There's specific mutation, this KIT D816V mutation. Unfortunately, most tyrosine kinase inhibitors do not inhibit that particular mutation. Midostaurin is one that does, but has some limited efficacy and a lot of gastrointestinal toxicity. Still a big step forward. BLU-285 is a potent selective inhibitor of the mutant form of KIT, specifically targeting the D816V, which is mutated in about 90% of patients with systemic mastocytosis. And so it lends itself to try and see what the efficacy is of this particular agent is in this disease. There are other diseases with the mutation, but my focus was on systemic mastocytosis.
So what did you do?
So it's a phase 1 study, and so the first thing we try to do is establish a maximum tolerated dose and establish a recommended phase 2 dose moving forward. So we started at very low doses, 30 milligrams a day. And the nice thing about this drug is it's a single daily dose, so it's very easy to administer. Started at 30 and worked our way up to 400. And we never established a maximum tolerated dose because the drug is very well tolerated. But 300 milligrams seemed to be well beyond what was needed to saturate and inhibit the mutant receptor. And so that was the dose that we went forward with.
And how many people?
The phase 1 dose escalation is 32 patients. Right now we're accruing into the part 2, the dose expansion cohorts, in specific histologic subtypes of systemic mastocytosis. Systemic mastocytosis is a complicated disease. Represents a multitude of different histologic subtypes, including aggressive systemic mastocytosis with associated other hematologic neoplasia, and mast cell leukemia. And so we're expanding cohorts of patients in each of those 3 disease sets.
What have you found so far?
So the phase 1, the part 1 of the dose escalation, which is really designed to find a dose, we were surprised that we had huge efficacy. First of all, of the 32 patients that enrolled on the study, 30 patients remained on study, arguing that A, the drug's well tolerated, and B, it seems to be efficacious. In addition, all 32 patients have responded based on reduction of the serum tryptase. Of the 25 patients who went on study with a lot of bone marrow disease, more than 60% of them have been able to establish a complete remission within the marrow cavity itself. So although the part 1 dose escalation was not designed for an efficacy endpoint, we saw huge amounts of efficacy in a very well tolerated fashion.
What about toxicities?
Yeah, so the toxicities of BLU-285 were very mild. Most were what we call grade 1, grade 2, mild GI, mild edema. The most common non-hematologic grade 3 toxicities were periorbital edema, swelling around the eyes, as well as fatigue. But that was mitigated by maybe a dose break and then reduction in the dose. No patient on the part 1 dose escalation of the study came off study for toxicity, which is a little unusual. So just arguing how well tolerated it was.
So the conclusions so far?
So far, we have a recommended phase 2 dose. We have a established pharmacokinetic steady state data. We've established that the target's a valid target, the D816V. We've established that the dosing level that we're at is able to inhibit the mutation form. And we've established that there's huge efficacy in a variety of histologic subtypes of systemic mastocytosis. So the plan right now is to confirm that with the dose expansion at the very specific histologic subtypes. And there are plans next year to more formally test the efficacy question in advanced systemic mastocytosis, but also, because this is a very tolerated agent, to look at more indolent forms, indolent systemic mastocytosis, as well as something called smoldering. And that's because the drug's very well tolerated.
You mentioned that this applies to 90% of the people with it.
So 90% of patients will have the D816V mutation. There are other mutations that are also inhibited by this particular molecule, the BLU-285. It's because it's so rare, we don't know what all the mutations, but most of the mutations, 90% of, or most of the patients, 90% of them will have this specific mutation.
Mm-hmm. And are people looking at the 10% at all?
Always looking at the 10%. There's always the odd man out.
No one's looking into that.
Well, I mean, it's a rare subtype. So of the patients who do not have the D816V, there are other mutations, D816Y, and other things that are also inhibited by BLU-285. When it's a one-off, it's hard to describe efficacy. It's all 1-1-1. Then there are those patients who have a wild type. This drug also hits the wild type form of KIT, but 90% versus 1 or 2% is very hard to establish confidence intervals about the efficacy of that. But we are looking into it, as well as other mutations. So these are not isolated mutations. So there's a panoply of other mutations that also co-migrate with the KIT, and looking at which of those mutations render efficacy or reduced efficacy is very important. But those are ongoing clinical questions.